Yeah. Well, highly esteemed audience. It's a pleasure to be here. What I will talk about is work by a colleague of mine, Maayke Stomp. I was also involved in a couple of others, Hans Matthijs and Lucas Staal. It's work that we have published already quite some time ago in 2007, but that also kept us thinking, and I thought it's really exciting work, so I'll present it to you. We have some new things, but mostly it's already available. But I think it's a very exciting topic to think about what is the origin of the spectral niches of phototrophic microorganisms. So if you take a sample from a lake or the ocean, and if you look into that sample, you'll find a very rich diversity of different plankton species. This is just taken from the canals of Amsterdam, a bit concentrated. And even if you're not a plankton expert, you already see by eye that there are dozens of different species in this sample here. Now this is quite curious, because if you think about it, the aquatic environment, the lakes are just rather homogeneously mixed phytoplankton with only a few limiting resources, nitrogen, phosphorus, light perhaps. So only a handful of resources, and if you would look at classical competition theory, you would expect only a handful of species, the best competitors for these resources, to survive. So why do we get so many different species? That's a classic question, and this classic question is known as the paradox of the plankton. It was first posed by George Evelyn Hutchinson in 1961. So why do we have this species diversity? Well, one classic approach to think about this is to think in terms of niche differentiation. So rather than the neutral theory that we talked about last week, I will talk about niche differentiation here today. And of course, the most famous example is the niche differentiation by Darwin's finches, where you have seeds of different sizes, and the birds with the small beaks eat the small seeds, and the birds with the large beaks eat the large seeds. And in this way, they avoid strong competition. So there's niche differentiation. We also took a boat, not to the Galapagos Islands, but in our case to the Baltic Sea. And there we investigated the Picocyanobacteria. Picocyanobacteria, as the name already suggests, they are the smallest cyanobacteria. They were discovered only in 1979, but we now know that Picocyanobacteria are actually the most abundant plants on our planet. They are highly dominant in the oceans. And you also find them in many coastal waters. We went for the Baltic Sea because the Baltic Sea is an interesting sea. It has this brackish system, and there had not been that much research on Picocyanobacteria in the Baltic yet. So what we found to our surprise is that there's a very rich diversity of different Picocyanobacteria there, and that all the strains that we isolated differed in color. So these are different strains that we isolated from the Baltic, and as you can see, it ranges from pink to dark green to light green to brown. So there's a wide variety of different colors of Picocyanobacteria. And why would that be? What explains this colorful diversity? Now, if you think about colors, then Newton has made an important contribution here. So I'm happy that we're now at the Newton Institute, that I can present this slide here, because Newton was the person who actually discovered that white light contains many different colors. Before him, people thought that white light was colorless, and that you had to add different hues of green or blue or red to obtain color. But he discovered with his prisms that we have a light spectrum, and that white light actually contains all of these colors, and it is described in his famous book Optics, his second book after the Principia, after the description of the Newton's laws in the Principia. So our hypothesis is, well, if white light contains many different colors, you can also have niche differentiation in the light spectrum. So different phytoplankton species, or in our case different Picocyanobacteria, can absorb different parts of the light spectrum. These are absorption spectra, and the peaks indicate where these organisms absorb most of their photons. For instance, this red cyanobacterium over here, it absorbs very strong in the green and the yellow, but it does not absorb much in the red and orange, and therefore it has a red color. If you look at the culture, it has a red color because it does not absorb red very efficiently. It absorbs green very efficiently. So what we did is we developed a mathematical model which included the underwater light spectrum. This is light intensity as a function of wavelength and a function of depth. It is described by Lambert-Beer's law, and this law basically states that there's an exponential decline of light intensity with depth. This is the light at the surface, I in, as a function of wavelength. This is light absorption by different phytoplankton species, numbered 1 to n. This is light absorption by what we call the background turbidity, so the light absorption by dissolved organic matter and by the water column itself. Then we can calculate the number of absorbed photons by the different species. So for each species we take this integral from 400 to 700 nanometers. That's the PAR range, the range in which they do photosynthesis. Over the amount of light that they have absorbed, as indicated by this equation. And then we say that the population dynamics is governed by this light availability. So the number of absorbed photons translates via a quantum efficiency in a certain production of new biomass. And this term here describes the losses of the biomass. So this is our system. And if we have 5 different species, then we have 5 of these equations, right? So each species is given its own equation to describe how its light absorption spectrum looks like, how it utilizes the underwater light spectrum. Okay, we first did a laboratory test, 2 different strains, isolated from the Baltic Sea strain 4 and Baltic Sea strain 5. This one contained a lot of phycocyanin, that's this pigment over here, it absorbs at about 640 nanometers. And this one had a lot of phycoerythrin, it absorbs strongly at about 560 nanometers. So they differ clearly, the pigment composition, as you can see in the color.
Why don't you get red plants?
Sorry?
Just curious, why don't you get red plants?
Yeah, that's a good question. Why are all these plants, why do they have only green leaves and only use chlorophyll? I don't know the answer. Except from an evolutionary perspective, we know that all the terrestrial plants evolved from just a few strains of green algae, right? They only came from one particular...
That leaves an enormous niche for several hundred million.
Yeah, you would say so, yeah. Yeah.
But chlorophyll is 650, right?
680.
680.
So chlorophyll is a bit
nanometers.
like
So this is chlorophyll. So this is phycoerythrin, but these 2 peaks are from chlorophyll A. It has this peak at 680 nanometers, and it is this peak known as the Sept band, somewhere around 450 nanometers. And also this guy here also has the chlorophyll peaks, both, and then the phycocyanin. Yeah.
Is it possible because near 700 nanometers, the energy is larger?
So the energy is highest, of course, in the blue, and if you go to the red, it gets lower and lower.
Okay, I see. Okay.
And basically, what these organisms do is that they catch the light. Here in this case with phycoerythrin, in this case with phycocyanin,
Okay.
and then they pass it on to the reaction center to chlorophyll at 680 nanometers, and this passes it on to the reaction center of photosystem 1 and photosystem 2.
So they absorb that band, as a result, the color is negative of that band. That makes
Sorry, they absorb in this band, yeah, and then?
Yeah, so we're trying to figure out, so it's red,
Yeah.
on the bottom is red. Is that because it absorbs of the yellow color?
Yeah, it absorbs. This is the red. This part of the spectrum is this part of the spectrum that absorbs the least,
Okay.
and that gives it its color, because that's the light that remains. Yeah, yeah. And here, this guy here absorbs the least in this part of the spectrum, and that's the green-yellowish light.
Okay.
So that's why it has this green-yellow,
I see.
yeah, color.
Okay, but so you're saying that a regular land plant wouldn't have the second peak?
A regular.
Land plant.
A land plant wouldn't have phycocyanin. It would have the chlorophyll A, so this peak and this peak. And then in addition, a land plant also has chlorophyll B.
Oh, okay.
These guys have no chlorophyll B, at least not the real cyanobacteria. Chlorophyll B is rather close to chlorophyll A. It has a peak here, a shoulder here, and it has a shoulder here next to 680. Yeah. So we did competition experiments, first in colored environments. So we put a red filter here, we have lots of those in Amsterdam, and then a green filter here. This is competition in red light, and what you see in red light, the green species wins, right? Because the green species is good in absorbing red light. You have to think complementary all the time. And here in green light, the red species wins, because the red species, as you saw in the previous slide, the red species is good in absorbing green-yellow light. Okay, these are the data, the symbols. These lines are the model predictions. Not perfect, but at least we were in the right direction.
Did you give them the same amount of potential energy in the sense of that?
We gave them the same amount of photons over 400 to 700 nanometers.
The same number of photons, but not the same energy, because you've got different energy at each photon, at each wavelength, right?
Yes, yeah.
Okay.
But here's Einstein who plays a role, because Einstein got the Nobel Prize for the photoelectric effect, which says that one photon excites one electron. So in photosynthesis you count the number of photons rather than their energy. That's why you see always micromole photons per square meter per second.
It doesn't matter what color the photon is.
Exactly. Whether it gets a green photon or a blue photon, if it gets one photon, it can excite only one electron. Yeah. Yeah. But it is important that it's within the range of.
Because it has to be in.
It is important that it's in the range from 400 to 700 nanometers.
Yeah, it has to be.
Yeah.
In order to be in the right. Yeah, everything within those things is within the right energy.
Yeah, okay.
It's too far that way, it's too energetic.
And basically what happens, so if a photon is absorbed with this wavelength of 560 nanometers, then the energy of this photon is passed on to chlorophyll, and the difference in energy content between this wavelength and this wavelength is lost as heat, and a little bit of fluorescence. Okay, then competition in white light, put the two species together, and what happened there is that they coexisted. And these are again data and model predictions. The lines are the model predictions. Dots are the data. So we can really predict that they coexist because of partitioning of the light spectrum.
Why, Jeff, why did the red line decrease after peak?
Yeah, this is quite typical in many competition experiments, that you have some end result, right? So where it goes to. But if, for instance, you give one species a bit of a head start compared to the other, then it might be that this first species, well, initially goes up high, but later on the other species is kicking in.
But they're not competing for the same wavelength, do they?
No, but they overlap, right?
They overlap, okay.
If you look at the spectra, this is really how they overlap. So they overlap here, and they overlap here, but they're really different in these two peaks.
Okay.
Yeah. So they do it.
How many should coexist? When you just have two.
Yeah, we'll continue with
Okay.
the talk. It will become clearer. So
Is the answer in your shirt?
no, would be nice. So what next? Well, we thought, our next step was, let's have a look at different aquatic ecosystems in the world and see what the abundance of red and green picocyanobacteria is and how this relates to the underwater light spectrum. Because we saw that red light should select for green species and green light should select for red species and white light should contain them both, at least in our lab experiments. Let's see whether that's true also in the natural environment. So we sampled many interesting ecosystems all across the world, in total 70 aquatic ecosystems. And we measured the underwater light spectrum. And if you want to investigate the underwater light spectrum, then basically you have to know that there are only few properties that really determine this spectrum. The most important one is obviously H2O, just the water molecule. This is the absorption spectrum of the water molecule. So water absorbs very strongly in the red, water absorbs very little in the blue, and that's why pure water or oceanic water with very low stuff of anything else has a blue color, right? It absorbs a lot of red, but what is left over is blue. Another very important component in aquatic ecosystems is dissolved organic matter. And people in the optics literature, they don't call it DOM, they call it Gilvin for some reason. It's an old German word, Gilvin. And this is the absorption spectrum of Gilvin, or dissolved organic matter. It absorbs a lot in the blue, and then there's an exponential decay towards the red. Now this absorption spectrum of water is a given. But this absorption by Gilvin depends on the concentration of Gilvin, right? This curve will become very high if you throw in a lot of organic matter. This curve will be very low if you have very little organic matter.
But both depend on depth.
Yeah, and it can depend on depth also, yeah. Yeah, on depth in the water column, yeah. So if we have a look here at three different aquatic ecosystems, first the clear ocean. This is the light absorption spectrum of pure water, and this is the absorption by Gilvin in the water, because organic matter concentrations in the open ocean are very low. So what happens in the open ocean is that you have strong light absorption in the red part, it's indicated here, and what's penetrating deepest into the water column is blue light. Now if you look at coastal waters, and at many mesotrophic or oligotrophic lakes, you will find again the same absorption spectrum of water, but a higher concentration of dissolved organic matter. So now we have absorption in this part of the spectrum, absorption in this part of the spectrum, but we have relatively little absorption here in the green. So what you see is that in these coastal waters and also many lakes, the green light dominates. The blue light is absorbed by the DOM, or Gilvin, and the red light is absorbed by water itself, but remains as green light, and the light doesn't penetrate that deep, say about 15 meters or so in this example. Now we can go to a peat lake, extremely rich in organic matter, that's indicated by this curve here, a lot of absorption by dissolved organic matter. So we have a very murky brown lake. What you will see there is that, well, this organic matter absorbs everything in the blue and the green, and the color that dominates is red light, but it doesn't penetrate very deeply. So in peat lakes you always have this red-brown color, and well, you can see up to perhaps a few decimeters depth, but that's about it.
You see a switch to cyanobacteria?
I'll show you in a minute.
You're good, okay.
So what we did is we sampled our 70 aquatic ecosystems, going from clear lakes and oceans, where the underwater color is prevailingly blue, towards very turbid lakes, where the underwater column becomes murky brown reddish. And this is the change in the relative abundance of the red picocyanobacteria, they decline, and the green picocyanobacteria, they go up. Each dot here is a single lake or aquatic ecosystem. Where we found the highest phytoplankton biomass. So in some systems just near the surface, but for instance in the open oceans there can be a deep chlorophyll maximum. Then we sampled at the deep chlorophyll maximum where we found the highest biomass.
So help me with the greens and the reds. So what's the name of the greens?
The greens, in some lakes it's microcystis. In other lakes, it's Aphanizomenon. In other lakes, it's green Synechococcus. That's the green cyanobacteria. The reds are very often red picocyanobacteria. The ones that they're all the cyanobacteria that contain phycoerythrin.
Right, which are what?
Many Synechococcus species, Trichodesmium also contains phycoerythrin. Some Anabina strains contain phycoerythrin. Glotrichia can contain phycoerythrin. Yeah.
How did you measure the light absorption in the field?
By Gilvin, what you do is you filter off the phytoplankton and all particulate matter, and you know the light absorption spectrum by water, and the rest is assumed to be Kelvin.
Oh, okay.
Yeah, so it's the only unknown
Okay.
in an equation. Yeah, and you just put it in a spectrophotometer.
So there might be a more explanatory variable. Because you're sampling at different depths.
Yeah.
Then you have, it's not just the Kelvin, it's also the depth. So there might be a way of measuring like relative light intensity.
Yeah, there will be more things going on than just this, definitely. Yeah.
You're saying that in the extremes.
And
So-
you focus on these two different types of cells, right? One
Yeah,
is that one.
because phycoerythrin and phycocyanin are key pigments in cyanobacteria.
Okay. Is there anybody else?
Yes, and I will show you.
Oh, okay, okay.
I will show you later. There are others.
And do you have
Good
any
question.
fill, can you give me a fill of Kelvin and micromolar GOM? Any fill?
Not out of my head.
Okay,
No, sorry.
all right, no worries.
So red species dominate in clear water, green in turbid water, and our Baltic Sea is right here in this middle region. That's the typical light absorption by organic matter in the Baltic Sea is in this range. We had the measurements also. So that explains, in our view, why we had such a colorful diversity of greens and reds in the Baltic Sea.
Wouldn't you
They
expect us to
coexist.
get yellows in the Baltic Sea? I mean, so why,
Well, no, they affect each other.
Distribution.
They affect each other's spectrum.
They affect the underwater light spectrum, yeah. So the
So
okay.
they take each other's leftovers.
Something happened to your x-axis, it's all messed up.
Oh, it's in Dutch.
oh, sorry. Very good. Yeah. Organische stof. I tried to pronounce it. Yeah.
Organic stuff.
So this raised some questions like, okay, that's nice that we find this relationship between the underwater light spectrum and the pigment composition of the phototrophic organisms of the cyanobacteria. But then let's think a bit further about what kind of niches are available in the underwater light spectrum. Is it like a continuum that we have here on the x-axis the wavelength from 400 to 700 nanometers, and then these are different aquatic environments and they can just be located anywhere along this spectrum? Or do we have some constraints on the niche space? And that's where the water molecules kick in. Because if you look at a water molecule can do three types of vibrations. They are known as the symmetric stretch. That means here you have the oxygen atom and here you have the hydrogen atoms. It's doing this. Right? Then we have the bending vibration. It's this vibration. And it vibrates because it's warm. There's a temperature and these molecules vibrate with temperature. And it can do the asymmetric stretch, so this movement. And these movements occur at particular energy levels. So here you see the absorption spectrum of pure water. In the previous graphs we looked only at the 400 to 700 nanometers range, but now I look at a much wider range of wavelengths. This is the absorption spectrum of pure water. And what you see here is that, for instance, this bending vibration requires the energy that's equal to a photon with a frequency of 1600 per centimeter. In other words, this V2, here this V2 level. So if it absorbs a photon at this level, then it can make this movement. So in a way, water prefers to absorb photons that match its energy requirements, so there's a peak in the spectrum at this wavelength. There's also a peak in the spectrum at these wavelengths of V1 and V3. V1 and V3 are very similar in energy content. So they require a bit more energy, these movements, these two stretches. And the fundamental frequencies are located here, 3000 per centimeter, or also about 3000 nanometers. So these are called the fundamental frequencies of the vibrations of the water molecule. But then a water molecule can also do combinations. For instance, if it absorbs a photon with this particular wavelength, it can do two movements. It can do a V2 bending movement and a V3 stretch. It's called a second harmonics. This peak in the spectrum is known as the second harmonics of the absorption by the water molecule. There's also another second harmonics, which links these two vibrations, two stretching vibrations over here. Then we have a third harmonics over here, with three combining in one photon yields three different movements of the water molecule, three different vibrations. And if we now zoom in further towards the visible part of the light spectrum, so now the graph runs from 400 to 1100 nanometers, we see again the two third harmonics. There's also a fourth harmonics. One photon leads to four movements of the water molecule, a fifth harmonics, a sixth harmonics, and a seventh harmonics. Right? So this explains the absorption spectrum of pure water. And you see some of these move into the visible spectrum. Now let's look at underwater light spectra again, a bit closer. So here we have again a clear ocean, coastal water, and a peat lake. And what you will see at the surface is that you have nearly white light at the surface. Usually a bit of the blue is taken away by Rayleigh scattering, if you're familiar with this. This is why our skies are blue, because there's a lot of scattering at shorter wavelengths. And then if you go deeper and deeper into the water column, then the absorption by pure water takes away a lot of the red light, and at 100 meters depth you find the dominance of blue light. This is our clear ocean water. This is again our coastal water. If at the surface it's relatively white, a little less blue. If you go deeper and deeper, it narrows down to the green wavelengths. Right? And here in a peat lake, if you go deeper and deeper into the water column, these are real measurements actually, you will narrow down to the red-brownish wavelength. Now what we can do is we can measure the underwater light spectra of a wide range of different aquatic ecosystems, and that's what we did. And here you see examples from 7 different aquatic ecosystems. This is the underwater light spectrum of the Pacific Ocean, measured at the euphodic depth. That's the depth to which 1% of the surface irradiance penetrates.
So that means you just kind of go to that depth
And then
shine a light meter.
yeah, and then measure the underwater light spectrum. Yeah, so in the open ocean it's about 100 meters depth or so. It can be 80 or 120, but about 100 meters depth you find this underwater light spectrum. So blue light strongly dominates. These are 2 lakes, this is the Baltic Sea, and this is a lake in the Netherlands, Lake IJsselmeer. It's a relatively clear water lake. Then here we have more turbid lakes with higher concentrations of dissolved organic matter. One is called Grote Moost, try it. And the other one is called Hilderpeel. And what you see also is nice peaks here. You see also that, for instance, this Hilderpeel has 2 different peaks in the underwater light spectrum, and also this Grote Moost has not a single peak, but has 2 different peaks. And this here is the most turbid system, and this is actually a mat. It's a mat in Yellowstone National Park with just half a centimeter of water on top of it. You can see this spectrum over here.
And the Yellowstone Lake is a lot of DOM inside.
Yeah, this one has a lot of DOM. It's a very productive net. Yeah. So what we see here are that we have some peaks and we also have some gaps in the underwater light spectrum. Well, you might think, well, measure a couple of lakes more and you will fill up all these gaps, right? But what we did is we have many more of these measurements and the gaps always persisted. So we thought, let's make a model and calculate the underwater light spectra using the absorption spectrum of pure water and adding different concentrations of dissolved organic matter. And this is an overlay of 100 calculated underwater light spectra, each time at the euphodic depth. So what you see is we have here one spectrum is a line, and then there's another line taking perhaps this peak, and there's another line over there. So we have a model that predicts us 100 underwater light spectra. We can make, if we do 1,000, we get the same graph.
So Jeff, this
Yeah.
is just water absorption and UM absorption?
Yeah. No phytoplankton, it's only water and DOM.
But that's not, that's different from what you showed to us, right? You had a very smooth lines, the diagrams, smooth diagrams. Or you just try to misguide us at the beginning?
So these are absorption spectra.
Okay.
This is how much is absorbed per meter,
Oh,
right?
okay.
And this is the light intensity, the irradiance of different
Sorry about
colors.
that.
Yeah.
All right, okay.
So yeah, I should be clearer. This is the light spectrum. The other one was an absorption spectrum. This is an these are absorption spectra, and these are light spectra, what you see as a result of the absorption.
Okay.
And these are measured light spectra. So what you see at this depth, you measure this at 100 meters depth, you find a lot of blue light.
So you get this because of the absorption spectra of the stuff above you, basically?
Yeah, yeah. You get this because of the absorption by water and by dissolved organic
What the
matter.
sun is in the first place.
Yeah.
And there's no biology goes into this?
No, this is pure the
Wow.
H2O and the dissolved organic matter. And what we see are again gaps and at the same places as in our measurements. And if we now look at the vibrations of the water molecule, here are the third harmonics, the two third harmonics, the fourth harmonic, the fifth, the sixth, and the seventh. These are these harmonics, these small peaks in the absorption spectrum of pure water, correspond with these large gaps in underwater light spectrum. Right?
So what do the harmonics represent? I mean, I know what they are, but what are they representing? Like, what's the explanation?
So the harmonics are the vibrations of the water molecule, and water molecules preferentially absorb photons with which they can release their energy because they're heated up, they want to move, so they preferentially absorb. They are better in absorbing light of this wavelength than of this wavelength or
I see. So
This
they're
wavelength.
catching it there,
Very little.
Yeah.
and then they're releasing it as heat. You would
Yeah.
see it as like,
that's their movement, that's the heat. Yeah.
so you'd see vibrational or
You see, it's vibrations of the water molecule. That's it. Basically, you give them energy in the form of light, and if they absorb a photon, they start dancing around, vibrating. Okay, so what this is telling us that it's very difficult to find an aquatic ecosystem where the light with 600 nanometers is dominant. Right? But it's much more easy to find aquatic ecosystems where light with 550 nanometers is dominant. So then also to understand the explanation, it's a bit strange. You have these very small, because these are just small bumps in the absorption spectrum, right? This is the line is going up here, and these vibrations of the water molecule just cause small extra bumps.
So you're down to 1%.
Yeah, but we go down to depth, and the reason is, in Lambert-Beer's law, we have an exponential decline of light intensity with depth. Now, if you have, and this is just a hypothetical case, suppose this would be an absorption spectrum of a certain substance, with a small bump in here. Then if you would look at this is absorption spectrum, then if you look at the light spectra with depth, then at the surface it's just pure white. If you go 1 meter deep, you see there's a little bit more absorbed here at this wavelength because the light absorption is higher. If you go further to 10 meters depth, this is already a deeper gap. And if you go to 100 meters depth, it's again a deeper gap. And that's because light intensity decays exponentially with depth, so that means that also small differences are enlarged exponentially. So that's why small bumps in the absorption spectrum of pure water, and this is on a log scale, on a linear scale. I do have the linear scale. Here, these are just the small bumps, correspond with the vibrations of the water molecule. These small bumps are enlarged to deep gaps in the underwater light spectra.
So does that mean if you sample the phytoplankton at different depths, you should get a completely different, because Because at the deep depth you've got niches basically.
Yeah. Now let's look further at our photosynthetic organisms. So we have seen that the top 6 panels we have already seen, right? So these are absorption spectra of water, pure water, and Gilvin or DOM. This is the underwater light spectrum from the surface towards 100 meter depth in the clear ocean, from the surface to 70 meter depth in a coastal water, from the surface to less than a meter depth in a peat lake. Then this is the irradiance at the euphotic depth. So this is actually it's this curve here, but then just the y-axis zoomed out, so then blows up a bit, get this curve. And this is the absorption spectrum of the dominant organism there. That's Prochlorococcus. So this is the light that's available at 120 meters depth. And you see that Prochlorococcus, it really loves to absorb light at this wavelength. Right? It has tuned its pigments to the available underwater light spectrum. The same in the Baltic Sea. In the Baltic Sea, if we look at the samples where we had a lot of red cyanobacteria, this is the underwater light spectrum here with a peak in the green, and these red cyanobacteria have their phycoerythrin just right in the middle of this peak. Right? And now we have the green cyanobacteria. It's not as nice. If we enlarge this graph, it looks a bit more wobbly than their actual spectra, but you can see that their spectra are tuned to the available light. So it makes more sense in this water environment to make phycocyanin, this pigment, than to make phycoerythrin. Okay. Now let's zoom in. Yeah.
If you don't mind. So H and I, that makes sense. Have peaks match the dark line.
Uh-huh.
But if you grow green, the I and H in G, they will have a chance. Will they have a chance to survive because they?
This one?
Yes.
But it would have a chance to survive here because of its chlorophyll A peak? That's your question?
Yes or no?
Yeah? Yes, it would have a
Oh,
chance.
okay.
Yeah. There are other reasons why it would not have a chance, because it doesn't always use, but it makes the story more complicated. But I will skip that part.
Okay.
Let's have a closer look now at the cyanobacteria, and we will look at the absorption spectra of different strains of Xenococcus. They're all marine strains here, 5 different strains. It's nicely shown. It's from a paper of Six et al. With David Scanlon from the University of Warwick. So they have these 5 different strains, and these are the absorption spectra of the 5 different strains. And you can see there are actually 5 different peaks here. They're known as, we have already talked about it, chlorophyll A here, absorbing in the blue at about 450 and absorbing in the red at about 680 nanometers. We've talked already about phycocyanin, this peak at about 640 nanometers. We've talked about phycoerythrin, but actually there are multiple types of phycoerythrin. One here, this one, is called PEP, or phycoerythrobilin. It absorbs mainly at 560 nanometers. And there's another one known as PUP, phycoeurobilin, and this pigment, or chromophore I should call it, but let's call it just pigment. This pigment absorbs mainly at about 500 nanometers. And if you look very carefully, you can also see that for PEP there can be differences. There are also different types within the PEP. This one has its peak at a slightly different wavelength than this strain. So these are the key pigments that you will find in the cyanobacteria. And many cyanobacteria, they almost always have chlorophyll A, and then they combine it with one or a few of these other pigments. And the combinations of these different pigments give them the different colors. Now the other famous organism in the oceans is Prochlorococcus, also a kind of cyanobacterium but with a totally different pigmentation. So this Prochlorococcus, I don't know whether you've ever heard of it, but it's the most abundant photosynthetic organism on our planet. We now know it's the dominant species in the tropics and the subtropics. And basically, there are a lot of different strains, but basically they can be distinguished in 2 different ecotypes, high light adapted ecotypes and low light adapted ecotypes. These are the absorption spectra. They don't have any phycocyanin, they don't have any phycoerythrin, but they have chlorophyll A, that's this peak here and this peak. And in addition, and that's new, they also have chlorophyll B, like the land plants, like the terrestrial plants. That's why these guys, they're prokaryotes, they're called prochlorophytes. Also, Prochlorococcus, it's thought to be, there's a lot of discussion about it, but it might be the precursor of the pigments in green algae and land plants. So here what is important for this story here is that we have a peak here of diphenyl chlorophyll A and here a peak of diphenyl chlorophyll B. Okay, now let's add the pieces of the puzzle together. Here we have the absorption spectrum of pure water, with its harmonics in the range from 400 to 800 nanometers, the fourth harmonics, the fifth harmonics, the sixth and the seventh harmonics. Also two what they call combination modes. This 4.1 is, I think, a couple of these stretches and plus one bending. This 5.1 is also a couple of 5 of these different stretches and then one bending. Then you get the combination modes for the vibrations. This gives rise to a range of underwater light spectra with gaps at the 7th harmonics, the 6th harmonic, a minor gap here at 5.1, a deep gap at 5, a minor gap at 4.1 and a deep gap at 4. And now I'll make an overlay of the pigments that we have just seen, of the cyanobacteria and Prochlorococcus. Right, just follow the lines here, from 7 down. And what you will see is that chlorophyll A has its niche below the 7th harmonics of the water molecule. Chlorophyll B, that's this one here, and PUB, phycoerythrobutylene, has its niche between the 7th and the 6th harmonics. Phycoerythrobutylene, the other phycoerythrin, has its niche between the 6th and the 5th harmonics, and even the different types of PEP are just on the left and the right hand side of this 5.1. You can see that the valley here, the separation between PUB and PEP the valley is right here where we have the 6th harmonics. The valley between chlorophyll A and B is right here at the 7th harmonics. The valley between the phycoerythrins and phycocyanin is right here at the 5th harmonics. The niche here is between 5 and 4.1 for phycocyanin. And the niche here for chlorophyll is on the right hand side of this 4.1. So basically, the whole picture fits. Our photosynthetic pigments fill in the niches that they find in the underwater light environments of our planet. Okay, this is up to, we talked about cyanobacteria, up to about 700 nanometers, because these cyanobacteria don't photosynthesize at wavelengths beyond 700 nanometers. But there are other microorganisms, anoxygenic, that do anoxygenic photosynthesis, and that can absorb light at longer wavelengths with lower energy contents. Examples are the green sulfur bacteria. They absorb light at about 720 nanometers. The purple sulfur bacteria, they absorb light at about 820 nanometers, the different strains. And the purple non-sulfur bacteria, they're also in this region here, so between 800 and 900 nanometers, and there are few species that can even absorb photons of only 1000 nanometers, a very low energy content, but they can still do anoxygenic photosynthesis with it. And then all these numbers here are different strains, different species. These are the cyanobacteria that we've seen before. These are eukaryotic phytoplankton with also characteristic peaks. And what I do again, I make an overlay of all these different absorption spectra. So I sum them all up, and this is the total overlay of all absorption spectra of the different species. You see again this landscape of hills and valleys. So we can do again the same trick. So in the previous case, we looked only up to about 700 nanometers. Now we look beyond 700 nanometers, and you see again. Now we have the 3rd and the 4th harmonics. You see strong gaps in the underwater light spectra, and they're reflected also in strong gaps. There are no photosynthetic pigments in these areas. So also the bacterial chlorophylls are adapted to like what is available in the underwater light environment.
Okay.
Yeah?
So, but compare that with a terrestrial environment. There's single light spectrum.
Yeah, it's so poor in interest. They have only chlorophyll A and B,
Wouldn't you
and
expect
this is
to have
They're always
much
out of
more?
CO2.
Well, I mean, in a shallow water, presumably you would expect much more diverse pigments.
yes.
Do you see that?
Well, let me phrase it in such a way. If you go very deep to a region where you have a very narrow wavelength,
Yeah.
then you see that only those organisms that have pigments in those wavelengths can survive.
Okay.
And on the
So you
surface-
might see lower or higher diversity near the surface, but
Yeah.
if you're in a red environment, you need to have the pigments that
Yeah.
can absorb red light.
Yeah.
So the diversity drops as you go deep.
Yeah, but there are many exceptions. There are also examples where the diversity at the top is not high because of other reasons, other factors, right? I only focus on the light spectrum. But if you go deep and there's only one wavelength available, you should have pigments in this wavelength.
You choose a depth of like 1% of the surface radiation. If you go, say, 0.1% of radiation,
Yeah, we tried this actually, whether our results are robust. But basically what you do if you go, for instance, to 0.1% is that these gaps become even deeper.
yes.
But the position is just at the same place.
Mm.
And the position is not altered.
Right.
That's the same principle.
So then does that mean that everything that everywhere nature selects?
Yes, I would guess that there's has been a strong selective pressure on the photosynthetic pigment. Yeah,
Must be.
yeah, it must be. Yeah.
Where does rhodopsin sit?
That's interesting because the rhodopsins, you also have a whole suite of different rhodopsins with different spectra, and we haven't looked into detail about whether they would fit this story as well. So we have looked at the bacteriochlorophylls, but not at the rhodopsins. We have one paper with Odette Bejian where we suggest that it also fits the picture, but actually the data are not strong enough to really say so, because we do not have an overview of all the pigments that you can, all the absorption spectra that you can create with rhodopsins.
I have a question that's sort of unrelated, but I'm sure you have an answer to what I'll ask you.
Yeah.
So you showed that the anoxygenic photosynthesis, they're using the photons on the wavelengths on the right side.
Uh-huh.
Why is that?
Well, what you need is this. For oxygenic photosynthesis, we have the Z scheme of photosynthesis with photosystem 2 and photosystem 1.
Mm-hmm.
And you need these two photosystems if you want to push electrons from the splitting of water. So here is H2O is split into 2H plus, I should put it like this way, it's better. Plus O2 plus some electrons that flow upwards. So if you want to go from the splitting of water all the way up to the formation of NADPH2, you need two photosystems and a lot of energy in your photons, because there's a large gap in the redox potential of these two reactions.
Right.
The anoxygenic ones, for instance, they don't use H2O, but for instance they might reduce H2S, and H2S has its redox potential somewhere here, and you need only one step to go to the production of NADPH2. And this step is not that big, so you don't need a photon that of a very high energy content. You can do with a lower energy content of your photons.
Right.
That's why they can still utilize photons
Okay.
at higher wavelengths.
But the puzzle that I feel is, when the photosynthesis first appeared, if I remember correctly, it's all anoxygenic.
Yes, it's this one.
And these guys, the whole spectrum of light are available.
Yeah.
So I would imagine there is no reason not to use the high energy wavelength.
Yeah.
And later on, oxygenic appears.
Yeah.
And they use the shorter wavelengths because that's the most energetic ones.
Yeah.
Is that reason because the competition, so they get out of, they kill all those who sits in the.
That might be. I don't know.
Okay.
Let's have a closer look again at the absorption spectra of the anoxygenic
I'm sorry, did you finish talking? Don't
Phototrophs?
don't
No,
mean
no,
to,
it's okay. But if you look at, for instance, here at the green sulfur bacteria, they have this peak here at 720, but they also have again a peak in the blue part of the spectrum, right? And also these guys have also peak in the blue part of the spectrum. So it might again be that they do the same trick. I'm not sure because I'm not a specialist on the anoxygenic ones, but I guess they do the same trick as the oxygenic ones, is that they can absorb photons of these wavelengths, then pass it on to reaction centers that are active at these wavelengths, and the difference in energy might be lost as heat. That's at least how cyanobacteria and higher plants do it. So I guess it's the same for the anoxygenic phototrophs. Well, to wrap up my talk, I think its conclusions are pretty straightforward. So we have seen that the vibrations of the water molecule cause certain peaks in the absorption spectra of pure water, and this creates a series of distinct spectral niches. And these distinct spectral niches are taken by the phototrophic organisms on our planet. So this has played a key role in the ecology and evolution of the photosynthetic pigments.
So what you haven't done,
Tell me.
well, you haven't related this to within an individual lode. In your Baltic Sea, you have these different colours at different frequencies, and you haven't told. So you told us that you can predict where the peaks are based on this overall quantity of light, but you can't tell us how many of these peaks are going to be occupied in any location.
Do you have one
or
organism per peak, or do you have sort of coexistence?
Yeah, so what you can have, for instance, if we go back, you can have several different things. One is if you look at some lakes, actually you find two peaks. Remember this lake here, for instance, has two peaks, so I would expect two different photosynthetic pigments, either in two different species or combined.
But the Baltic
in
Sea,
one.
the
The Baltic Sea has only one.
It's got
Yeah,
millions of them.
so you would expect, you're completely right, you would expect here basically only the phycoerythrin cyanobacteria. And if we go into detail into our data set, you will see that if we are at the 1% light level at the euphoric depth, it's mostly the phycoerythrin containing cyanobacteria, but closer to the surface, it widens out, right? Because these are the data from the Baltic Sea on the water light spectrum. So at 17 meters, the only light available is green, and you need to have the pigment phycoerythrin to do something here, to do photosynthesis. But here at 3 meters depth, well, there's also red light available and blue light. So here there's a possibility for a wider range of photosynthetic organisms.
So what made you decide to go 1%?
Because it worked. It gave us like, okay, and 0.1% also
so you
worked.
work this out before
Huh?
you decide 1%. Did you see this graph before you make a decision go
You've
to?
got to decide.
No. We choose 1% because if you ask anybody working on phytoplankton and photosynthesis, like what is the light levels up to which you find active photosynthesis, they would say, oh, it's the euphoric depth at about 1%.
Okay.
So we thought, well, that's the most logical choice
Okay.
to look at. But if I would have made, if I would have made these graphs at the 10% light level, the landscape would still be hills and valleys, only this valley would be less deep.
Right. Then if the value is a little higher, then the competitive pressure will not be that.
Yeah, true.
Yeah.
So that's an interesting question. Then the competitive pressure would be less, but still we find this nice match.
But then they interact. So to what extent does it work? Well, you said that in the experiment. So one that you've shown that in experimental conditions that there is a steady state. Okay, but you haven't provided evidence that in the oceans there is the same steady state.
Oh, there's no steady state in the oceans. I don't believe in steady states. Only in my chemostat.
I think there's only steady states in the ocean.
Well, I mean, they don't have to be.
No,
so,
but it's just an attractor, right?
Yeah, an attractor.
Yeah.
You will see, I mean, if you go out at a higher depth and you measure frequencies of different things, and you average it over a large thing, you sort of have an attractor.
Yeah.
I don't
Yeah, yeah.
Okay, but that has multiple organisms in it.
Yeah,
And
with
the
different
reason-
light absorption spectra.
And that's because they do. What proportion of the absorption is actually done by organic?
Oh, that's an interesting question. If you look at the open ocean, the concentration of phytoplankton are very low, and they absorb perhaps only 0.1% of the light energy
So in that case, you
So-
should have a single wavelength.
at greater depth that dominates, right? At 100 meters depth, it's blue light, but close to the surface, it's a broader spectrum.
Okay, but the best. If they're not competing with each other,
Uh-huh.
then the best photosynthetic system should win.
And it should be the one with the blue, which can fill in this blue niche.
Well, that should also be true near the surface. Because even small photovoltaics will just make you really late.
Well, so I'm just asking.
No, well,
questions which-
at the surface, well, let's just do the simple game. We have done these experiments actually also, but at the surface you have a spectrum that maybe looks something like this. So if you have an organism that has its, just take a very simple organism that has only one pigment, one peak over here,
Yeah.
then basically this is the absorption spectrum, the light available that we observed. This organism comes in and starts growing, and what it does, it creates a gap
Right.
Yeah.
here in the light spectrum when it becomes abundant. It absorbs all this light. So then it becomes, then other species with absorption peaks here or here,
Right.
Yeah. So how important
be
is
failing.
that? So that isn't important right at the surface.
this is important at the surface, right? In white light, you'll find a lot of species that will fit into this white light spectrum.
But if you're right at the surface, then it doesn't matter because there are
That's
no.
true, because nothing's absorbed.
So
Okay.
if you're right
In,
sitting right on
in the
top-
limit. What happens in the limit at the surface, I don't know. But as soon as you go a little bit deeper,
So
a few centimeters, yeah.
So within some substantial proportion is absorbed by living organisms.
Yeah, and
some
in lakes you can have that about 40 or 50% of the light is absorbed by the organisms. Yeah. And we did experiments. I don't have the graphs. That's a pity. But we did experiments with a chromatic adapter. Some of these organisms can change the pigment composition very strongly. They have both the phycoerythrin and the phycocyanin, and they can turn from green into red, and from red into green. We did competition experiments. We had a green picocyanobacterium compete with an adapter. So when we had a green picocyanobacterium, then the adapter turned red. When we had a red picocyanobacterium, then the adapter turned green. So what they basically did is they take the complementary colors, because that's the niche where they can still
They
survive.
don't try to kill each other?
Hmm? Yeah.
They don't try to kill each other. So,
Sorry?
Okay,
Yeah.
good.
so your results makes me, I have a wild idea, but probably you already thought of it. So what I see the result you show is photosynthetic bacteria evolved from deep. They did
Yeah.
not evolve on the surface.
Yeah, or the selection pressure on average has been such that even though they were at the surface as well, the selection pressure acted
But the surface wouldn't.
when they.
The surface wouldn't have any pressure.
No, at the surface you wouldn't have expect.
That's
any-
right.
yeah,
So
yeah.
if the cyanobacteria evolve on the surface, you wouldn't be able to see that.
No, but if they evolve, so one thing might be they evolved only at depth, or they were mixed, and say during part of the time they saw only the blue color of the oceans,
Mhm.
during part of the time they saw the more whitish spectrum at the surface, and then because they were mixed, it's
Right,
still.
Right, but
Process selection.
there must have been some significant pressure so that they preserve this nice peak.
Yeah.
Over the history.
Isn't it very strange that?
But that's something I would, if there are people interested, that's something that one might try to model, right?
Yeah.
So we have this spectrum we have it going from white at the surface to
Yeah.
a specific wavelength at depth, and how strong are the selective pressures
Right.
to get this result?
Because there's a hypothesis in geology that there are competing hypotheses. where did life evolve? One is in the middle ocean region, deep, and then the other is, you know, the ocean is the soap of life. You know, it can evolve from surface. So this result is very, very interesting. Very, very interesting. Very nice.
But isn't it strange that no plant, I mean, even if all the plants came from cyanobacteria, a plant that finds a red one should surely have a huge...
Yeah, I completely don't understand why there are not
You could
red.
make a plant with
Yeah, I completely don't understand why there are no red plants. I don't know.
It's quite bizarre.
I thought plants were always CO2 limited.
Oh,
I thought
No, they
everything-
can't. That's
Yeah.
they can't. You can't
so
get
it
to
wouldn't
that.
be any advantage.
You can't.
because
If
you're
you're
getting-
if you're on the bottom of a forest.
Yeah, the understory of
You're
a forest.
CO2.
Yeah.
You're still CO2.
Why CO2 limited?
They also like
There's no CO2 in the atmosphere, right? It's less than.
But you can always make it dark enough so that they will become light limited, right? If the understory is dark enough, at some point.
I have a go, but I will catch you. Sorry later.
See you.
Okay, bye.
So I did, I mean, we can check, right, but that's my understanding is that the difference between the ocean and the land is that you are basically CO2 limited. It happens in the ocean too sometimes, but only in like bays and stuff.
Yeah, but I would guess in the understory, because people did experiments. There has been a famous Nature paper a couple of years ago by Andy Hector and colleagues. What they did is they had a grassland, and then they put also lamps in the soil to illuminate the vegetation from below,
Yeah,
and then they saw a strong increase in productivity.
yeah.
And that's because indeed at the surface, at the top of the vegetation, there's CO2 limited, but if you go deeper into the vegetation, it gets so dark. And
Yeah,
it
but that
shifts.
must be only where you've got like the heterotrophic activity must be really high, like right at that interface. Right, because they're always pushing up, right? I do know that you can always get more plant.
If you give more CO2, yeah.
Yeah,
Yeah.
when it comes to the
Yeah.
terrestrial
Yeah.
environment.
But
by
I
a
would-
lot.
But I would guess that the lower stories
At the lower stories, yeah.
are
I don't
like
know. There's no CO2 in, like in a regular forest, like down deep, there's not much
Mhm.
there.
But-
Like ferns and stuff, just, they're really good at, reason ferns win is because they're good at using CO2 as long as there's a lot of water around, right? It's not a light question for them.
Yeah, and because they make a very large leaf area index.
Exactly, and
Yeah,
big,
yeah.
big, whatever, the big holes for getting in
Yeah.
CO2. Stump, what is it called? Stump?
Huidmondjes in Dutch. Stomata.
Yeah, that sounds right.
Yeah.
And you can see that, the only reason I say that, you can also watch the geological record of CO2 just by looking at the stoma size in plants and plant plants.
Yeah, which is strongly correlated with
Yeah.
CO2 pressure in the atmosphere. Yeah, that's correct. Okay, so puzzle for the land plants is not solved.
We haven't done that.
But also, I mean, it seems like from all this information, you could make a model that predicts how many coexisting species you should have at each depth.
Yeah.
Assuming no other.
Yeah, at least how many pigment types you would expect at
Yeah,
each depth.
exactly.
Yeah,
So, I mean, why don't you do that?
yeah.
That's a nice thing to do.
It's good.
Well-
The next step is to look at assemblages, which you've kind of made a meta assemblage of everything.
Yeah.
Okay, but the next thing is how are these meta assemblages actually assembled
Yeah.
to make real assemblages?
Yeah, and do you get indeed limited subsets of
Yeah.
species or pigments in
And,
certain
is it, and
environments?
how
Yeah.
different is it
Yeah.
between the competitive regime and
And a surface regime.
or open notion?
Yeah.